Hello friends, welcome to EPG Pathshala. I am Dr. Samandar Kaushik from Center for Biotechnology, MD University Rohtak, Haryana. Today we are going to discuss about the modules Biosafety issue in the animal cell biotechnology under the paper Animal Cell Biotechnology. The main objective of these modules is what is biotechnology, what is containment levels, and classification of the biosafety levels. There are 4 biosafety levels known as biosafety level 1, biosafety level 2, biosafety level 3 and biosafety level 4. One is the lowest biosafety level 1, in which we are handle the less pathogenic microorganism, and biosafety level 4 is the maximum level of biocontainment levels, in which we are able to handle most dangerous pathogens. Biosafety is a discipline addressing the safety handling of the contaminations of the infectious microorganism and biohazardous substances. A biosafety level is a set of the biocontainment protection required for isolate of the dangerous biological agent into the laboratory's environment. There are the 4 different levels of the contaminant from lowest to highest is known as biosafety level 1, biosafety level 2, 3 and 4. Why the biosafety issue in animal cell biotechnology? It is very important issue. There is a great role of biosafety in the animal cell culture technology. First aim of a biosafety is laboratory safety to protect the cell material from the environment of the lab or infection from the other instrument which are previously utilized for any tissue culture procedure. Second, you can protect the lab personnel through blood-borne pathogen by applying the biosafety rule to avoid the infection in the lab personnel. Nowadays, recombinant DNA technology is one of the important subject, and the gene from one organism may be transferred to any other organism. So such type of experiment should be done strictly under the biosafety levels to avoid unnecessary transfer of these materials. Sometimes infection may be start from the waste material which are generated in the laboratories, and these waste material should be contaminated as per the biological rules under the biosafety regulations. The infectious and diagnostic specimens should be handled by the persons in the laboratories. They may cause the infection to the persons, so such a protection can be possible with the help of biosafety regulation. There are certain specimens which are infected with respiratory pathogen. For example, a sample for influenza viruses, a sample for Mycobacterium tuberculosis diagnosis. If the person handle these sample without biosafety rule and regulation, the persons may be infected from these samples. Therefore, these materials should be strictly under biosafety rules. Another aspects of the biosafety is to handle the situation in case of the bioterrorism. If there is a outbreak of a pathogen or utilized a pathogen as a bioterrorism, this can be decontaminated safely with the help of biosafety regulation. Occupational safety and health in the research laboratories can be performed with the help of biosafety issues. Biohazardous used in the animal model can be safely handled with the help of biosafety level. Which type of the biohazardous material may be present in the animal cell culture? There are a large variety of the microbial contamination may be possible in the animal cell culture. Most important of them are viruses, example influenza viruses, rabies viruses or any other type of viruses. may be present in the animal cell culture, because this cell is obtained from a organism. This organism may be infected from any type of pathogen. Another chance of contamination is the bacteria. Bacteria may spoil your cell culture, so it should be handled in biosafety cabinet. Another microorganism which may contaminate your cell line is the fungus, or another type of the atypical bacteria like chlamydia, rickettsia, which may be possible to infection in the cell lines. And another type of the microorganism known as prions, which may also present in the animal cell cultures. This most frequent type of the atypical bacteria which are found in the animal cell culture contaminated is mycoplasma. Because of its small size, it may be directly filtered through some membrane filters or any other compound. It cannot be destroyed through the general method of sterilization, so it may be contaminate the cell lines. The recombinant DNA may also present in some type of the cells. In case of cell lines, a specific type of the cell should be present in a specific type of the culture flask. If you are handling more than one type of cell culture and simultaneously, sometimes the cells from one serotype or cells from a specific type may be contaminated another type of the cells. For example, if a person handling MDCK, Viro, LLMCK2 or any other cell types, the LLMKC may be contaminated in MDCK or any other cell type. So this is also considered as the contamination of cell, and your result may be effective. Yes, we are discuss about the many way in which animal cells may be contaminated. Then what is the solution? How can you control the contaminations to safely handle of a animal cell culture with economical value? You can handle the cells in the different biosafety levels. And so now we are going to discuss these biosafety levels, and there are 4 different biosafety levels. They are typically known as biosafety level 1, biosafety level 2, biosafety level 3 and biosafety level 3. These biosafety levels provide the safe handling of biohazardous substances at a justified cost. These biosafety level increase the safety of the worker as well the environment from the biohazardous materials. And there is a specific guideline for working safely in the research or medical laboratories to safely handle of biohazardous materials. In case of the animal biosafety levels, these are known as animal biosafety level 1, 2, 3 and 4. The facility provided in the animal biosafety levels are safely handle of the laboratories animals, and the animals models that supports the research can be handled at a specific levels. There is a proper guideline for working and safely handle the animal for the research purpose should be followed before doing any type of experiment with animals. When you are working with any type of the microbial organism, you should follow some basic rules. This rule is known as General Good Laboratories Practice or General Good Laboratory Techniques. These are under the heading of no smoking. No one should be permitted for smoking in the laboratories. Eating. Sometimes eating may also responsible for the inoculation of microorganism through your food. So there should be no eating permit in the laboratories. Do not store the food in laboratory refrigerator, because this may cause the spoilage of food or contamination of the food, and which may cause the infection when some person consumed it. And applying the cosmetic, lip guard or any other beauty cream on the face or any other part of the body in laboratory should be prohibited, because this may cause some micro injury and pathogen may enter into the patient body through the skin. And restrict the entry into the lab. Lab is not a park, not a public place, so only authorized person should be enter into the laboratories. One of the best habit to protect the infection of the person, everyone should wash their hand before and after the procedure, which is very helpful assist to protect the environment and person from the infection. And one should follow the proper discard of the waste material, which is also very helpful to prevent the microbial contamination into the environment, and decontaminate the lab beds before and after the work. Journal good laboratory practice is also come along with the person should wear a proper clothing. For example, everyone should take the precaution or should use the personal protective equipment as per the requirement of pathogens. The minimum everyone should wear while handle with any type of microorganism, that is the lab coat, safety glasses and the gloves while working with any microorganisms. And you should plan your work first, and you should know in advance what you are working with. You should also read available resources supply and a material data sheet, which is a documents that contain the information of various potential hazardous substances which may be very dangerous to your health, which is flammable or not, and give the clue of the which react with any acid base or any specific chemicals, and give the guideline about how to discard these chemical into the environment. So everyone should read the MSDS sheet before handle a new chemicals. Standard microbiological practice. Most important rule is that everyone should follow the standard operating protocols. The awareness of potential hazards should be considered before handle any material into the laboratories. The laboratory staff should be trained and proficient to do any specific technique. And it is the moral responsibility of the laboratory supervisor to provide the appropriate laboratory facilities for the worker which are involved in any time of the biohazardous experiment. The worker should be trained in every respect, and special practice should be performed before doing any type of the hazardous experiment. If the person involved in the occupational health, then person should be trained in occupational health program through the higher institute which are considered as a specifically trained in the field. For example, if a person doing some viral experiment, should be trained with the higher institute like All India Institute of Medical Sciences, New Delhi, or only the institute which are deals with the virus, that is National Institute of Virology, Pune. They give the proper guidelines and they give the specific protocols to protect the personal health and to protect the environment from that biohazardous substances. Biosafety is one of the major issue while you are working with the animal cell culture or any other living systems. The biosafety can be maintained with the help of certain rule and regulation. For example, the entry should be restricted into animal cell culture. Only authorized person should be entered into the designated lab. The biosafety levels can be maintained with the help of certain instrument known as biosafety cabinet or laminar flow. This is the laminar flow which will produce the sterile air through these filters, and a sterile air will be blown toward the face of the worker. So you can perform only the sterile work in this and with non-infectious matter only. Another instrument for the biosafety is known as the biosafety cabinet. While you are working with non-infectious matter, laminar flow is the end. While you are working with the infectious component or infectious biological compound or patient sample or animal's material which may be infected by any virus, bacteria or any pathogen, that should be handled into biosafety cabinet. There are many categories of biosafety cabinet. This is biosafety levels to food which are designated for biosafety level 2 categories. In these categories, mainly those medically important pathogens are handled which are medically important, and there are some little bit prophylaxis available against them. Means those which have some infection and responsible for the infection into the persons, but there are some little treatment available against it. Or you have higher level of the biosafety cabinet available which are recommended for those pathogens which have no treatment and highly dangerous to the persons into the society. For example, for Mycobacterium tuberculosis or for HIV virus, we require some higher version biosafety levels. And another higher level is biosafety level 4 required the another type of the biosafety cabinet in negative pressures, because due to negative pressure one component or one things or one pathogen cannot enter into the environment. Anything came from the environment to the laboratories but cannot enter into the environment from the laboratories due to the high level of the positive pressure, high level of the negative pressure into the laboratories. And one should be protect themselves through the proper training of utilize the instrument as well as proper training of the personal protective equipment, which are very helpful to protect the person from get infection or also protect the environment for spreading the infections. Safety is also depend upon the practice doing by the personal staff and the equipment available in the laboratories and the design of the laboratories. All these in combined forms gives the a meaningful result to protect the person and environment from the infections. So that these can be classified as the primary containment barriers which are responsible to minimize the exposure of the person from the biohazardous substances, or they can prevent the persons by contact with the aerosol generated by these biohazardous substances. They can be engineering controlled or with the help of equipment. For example, your laboratories have a negative pressure, then the material which are handled in the laboratories cannot easily separate into the environment because of the negative pressures. If you are using the biosafety cabinet or laminar airflow, then the person doing the experiment on these biohazardous substances will be protected from the infection. And everyone should wear the personal protective equipment as per requirement of the nature of the microorganism which he was going to handle. These organisms should be handled as per the requirement of biosafety cabinets. And if you are involved in the animal experiment, there should be covered or ventilated cage system should be available for your experimentations. Secondary barriers mean engineering control. Your laboratory design should be such that air entry or out should be controlled, or contributions to the workers protection. They protect the laboratory from outside. The laboratory should be rat or insect free. The building and lab design is also very helpful in the protection of the environmental contamination. For example, infectious lab should be present in a separate building. If there is a shortage of building, these labs should be at least at a corner of the main building, and the lab should be provided with autoclaves and cage washing facilities. Now we are going to discuss different biosafety levels. Why we require the different biosafety levels? For example, if you are handling a simple microorganism like plasmodium, which are responsible for cause malaria only and cannot transmit from one person to another person without the help of mosquito, so you required only biosafety level 1 facilities, and you can perform the experiment on open table also. But if you are handling the most dangerous pathogen, for example rabies virus, for example Ebola virus, or any other viruses or pathogen which are not found in our countries, you may be infected from this pathogen, and it is very dangerous and responsible for the death of the person which are involved into handling of that such a dangerous pathogen. So you require to handle such a dangerous pathogen into different levels or maximum biosafety levels, that is biosafety level 4. So there we use the different biosafety level as per the requirement or nature of the pathogen that are going to be handled into the laboratory. So we are discussed the different type of biosafety levels. First of all, biosafety level 1, the organism which are well known characterized can be handled into BSL-1. The organism not to cause any human disease can be handled by the human being in these levels. There should be some primary treatment available for this pathogen, and you can perform your work on open bench if the organism is non-pathogenic for the human organism. For example, if you are working with E. coli, you can do the experimental procedure on open bench, or the organism which are generally not spread from material to the person. If you are doing the experiment with plasmodium, you can do you are testing the plasmodium, you can perform your experiment in open bench. And the animal in open cage system or open environment can be handled of such a situation when they are not contaminated with any pathogens. The good laboratory practice should be followed. The organism which can be handled into the biosafety level 1 are the example E. coli, transgenic plant, plasmids, fungus, mold, and certain yeast. Basic infrastructure should be required for biosafety level 1. You can perform the work at bench top, so there should be a bench, but you should decontaminate the waste material daily, and manual pipette is can be performed. Required handling, washing, and red bag should be present to discard waste material, and biosafety cabinet is not required. If you lab have, then it's good. Biosafety level 2. In case of biosafety level 2, the agent associated with normal human disease can be handled into BSL-2. The treatment of such a disease should be available. The agent should have a moderate hazardous to the personal and environment. The direct contact or exposure may be dangerous from these pathogen. The percutaneous exposure like scratching, pinching, or needle sticking may cause the infection into the persons, or when the mucous membrane are exposed, that is through eye, mouth, or open cut, this organism may cause the infection. So in such a situation when mucous membrane or there is a cut into the body, the person should avoid the work. The pathogen belong to the biosafety level 2 are generally herpes virus, all hepatitis virus, dengue virus, chikungunya viruses, and the others pathogen except the mycobacterium, other bacteria can be handled into these levels. For biosafety levels, one should precautions that the limited access to the laboratories while work is on progress. Daily contamination should be done after finishing the work. Mechanical pipetting should be advisable, and the lab personnel should wear the lab coat and safety glasses and gloves as per requirement. The lab bag should be present into the bag in the form of red bag, yellow bag and black bag, and the waste material should be discarded as per instruction of the hospital infection control policy, and the sharp container should be available to immediately cut the sharp objective which are utilizing into the lab. Biohazardous sign should be posted at the entrance of the laboratories. Labels are the equipment properly, and the tissue culture room should be negative airflow, because during the culture the amount of the pathogen is increased many times and they may cause the more infections chance when they contact into the susceptible persons. The documented training of the staff persons which are involving handle the infectious material in the biosafety level 2 laboratories. Baseline serology should be done, or pre-vaccine may be required if the vaccine is available against that pathogens, or especially for hepatitis B virus. The person should be vaccinated with the hepatitis B vaccine before doing the work. Then the next level is known as Biosafety level 3. The Biosafety level 3 is utilized to handle indigenous or exotic agent, that is which are not present in our area. The aerosol transmission is high for a pathogen can be handled into BSL-3 facilities. It is the facilities for those pathogens which are responsible for serious health effect on the persons. The treatment may or may not exist for these pathogens. The pathogen which belong to the biosafety level 3 is HIV virus or mycobacterium viruses or any other dangerous viruses of the equivalent capacity. Although HIV can be handled into BSL-2 facilities also, but it is advisable due to the image of these viruses should be handled into biosafety level 3. For biosafety level 3, one should take these precautions. Public access should not allowed at any cost into the laboratories. Daily contamination after the spillage of any materials or autoclave required at spot, so waste material is minimum transferred for decontaminate, and the no sharp should be used until absolutely necessary. Aerosol minimize with the procedure should be required, and the special equipment may be required for depending upon the procedure. Biohazardous sign and label should be posted at airflow control system, and documenting of the training and personal completely certification should be available in the laboratories. The baseline serology and vaccination is advisable for the biosafety level 3, and the spills report to the lab in charge immediately, and necessary action should be taken accordingly. And the standard operative protocols, biosafety manual, biosafety officer should be present in the laboratories. Biosafety level 4, that is the maximum contaminate facilities. The building on be available along with the biosafety level 3, and there is a integral part or additional part of the BSL-3 and BSL-4 should be present in the adjacent position with each other. The maximum contaminate facilities available for infectious disease is known as biosafety level 4. The pressurized containment should be wear while working the person in biosafety level 4. BSL-3 plus class 3 biosafety cabinet should be used, and the autoclave in the biosafety level 4 should be double-handed. From one end should be used for enter into the material, and other hand and should be utilized to discard the material after autoclave. The liquid effluent collect and decontaminate facilities as well. There is a no biosafety level 4 facility exists at MD universities, and only two biosafety level 4 laboratories available in India. One is at Bhopal, another biosafety level 4 laboratories available at National Institute of Virology, Pune. The virus which can be handled in biosafety level 4 are Ebola viruses, Lisa viruses, and the Marburg's viruses, or any other pathogen which are not found in our countries can be handled in biosafety level 4. For example, yellow fever, which are not found in our countries, can be easily handled in the biosafety level 4 facilities. The whole model can be conclude or summarize in a way. One should follow the biosafety levels. Different biosafety level required for the different level of the microorganism as per nature of microorganism, and there are 4 different levels of the biosafety levels. Main of the biosafety levels is to reduce the element or exposure of the laboratory's worker or any other person as well as environment to potentially dangerous agent which cause the infection into the person or which may contaminate the environment. Therefore, requirement of the biosafety depend upon the nature of pathogens to be handled in the labs. Thanks.